tissue except the skin,
Really?
skin and brain. Okay, so it's number 3. Secondly, virtually every human with an eye genetic disease is identified because the mother either sees it or the patient says, I can't see. And so ophthalmologists have been identifying genetic diseases of the eye, beginning really in Germany more than 110 years ago. So most of these diseases are now documented. And so there was a vast, therefore, reservoir of in genetic eye diseases where the affected gene wasn't known. Okay. Third reason was that in people and in animals there are a lot of animal models. For example, mice, where you look at the mouse and you say it's got no eyes or abnormal eyes or whatever. Right. So they're good models to study to get insight into the human diseases. And the last reason is the eyes. It's the only part of the nervous system that you can look at in the clinic if you're a doctor. When a doctor's looking in your eye with an ophthalmoscope, he's looking at your nervous system. Yeah. In fact, which people don't really, even doctors don't appreciate what that means fully. So, but it's a very extremely simple part of the nervous system. So it was simple to study the physiology of it, how the electricity and cells talk to each other. That was very well understood, but the actual molecules involved in its function and in these diseases were not identified. So there was a lot of background information, but few genes and therefore proteins that were mediated all this stuff I've been talking about were identified. So it seemed like in fact, only one gene had been identified. And so it seemed like a relatively open field. So then I had to think of an idea, okay, well, it's nice to say you want to find genes that are important to the retina, but how are you going to do that? You know? And so I spent 6 months thinking about that and ended up with a very simple strategy, which turned out to be a home run, even though the initial granting agencies to which I submitted this idea said, oh, this is a fishing expedition. And fortunately, I was at the Hospital for Sick Children, and this shows the importance of having really smart people behind you. So Lou Siminovich, and the head of the institute, said, you know, to heck with those funding agencies, just do the experiment. Because we did it and succeeded greatly. And then subsequently people would say, well, of course that idea would work, you know. Well,
what was the moment of discovery like? I mean, how did you feel exhilarated? How did you feel?
Yeah, I felt exhilarated the day that I had a postdoc. So we did this simple test of this this approach we thought would find us genes that were important to the retina before we knew what they did, which sounds like that's impossible. It's not impossible. And so I had a postdoctoral fellow working on this project, and she opened the door of my office one afternoon after they'd been at it for about 3 months, and she said, you know, one of these genes we're looking at looks like a gene that's important for development in flies. Do you think that's significant? You know, I nearly jumped out of my seat, and of course it was the kind of gene that's critical to developing all organisms in biology. And that turned out to be probably the most important gene we discovered. So it was cool.
And this was done where?
It was cool.
This was done where?
At Sick Kids in Toronto.
Yeah,
it was fun.
Was this when you approached the Howard Hughes Institute?
No, the Howard Hughes Institute approaches you. You don't approach them.
Okay, but you were appointed an international scholar.
Yeah, that's nice. That's a nice thing to have. Yeah.
And they were prepared to fund this particular.
Yeah, it's a good. They have an international scholar program, which is I think very sadly winding down. Very sadly, again for 2 reasons. One, Canadians are probably, this is, I think we'll all agree, the last group of citizens in the country to recognize the tall poppies within their population, unlike Americans, where they're always cheering their own. And so if you're an international research scholar of the Howard Hughes Medical Institute, the Canadian community finally recognizes maybe you're doing something useful. I mean, this is really true, I think, in all walks of Canadian life, not just science. But more seriously, and all joking aside, this International Research Scholars Program was fantastic for people from countries like Mexico and Argentina and Brazil, and we had scholars from there too, who were very, very capable, intelligent people who just didn't have the resources to support the research in their own country, and they got it from the Howard Hughes Institute. And so I worry about what's going to happen to people like that now that they're curtailing that program. I'm kind of mystified as to, Canadians don't need that program, but those countries need it to take their, and there's obviously remarkably capable people in all countries like that. And this gives them a chance to develop research in their own country. So it's unfortunate.
In 2009, the Toronto Star hailed your research on the protein NETO1.
Yes,
It was a collaborative effort at Sick Kids.
that's correct.
Now, was this a change in direction? I mean, you've been doing eye genetics. This is now synaptic genetics?
You must have to do a lot of work to do these interviews.
You have no idea.
I'm sorry. You know what you do in research is you go where the research takes you. So,
Right.
you know, throughout my career, I went from studying inherited biochemical diseases and then diseases of the retina, and then the reason we got into the area of the particular gene and protein you mentioned, NETO1, and there's NETO2, is because I had a brilliant young graduate, probably my most talented graduate student ever, started in my lab around 2000, David Ng. And we'd been working on a particular class of genes for about 10 years in my lab. The class of genes I alluded to earlier that was similar to the one in flies. And frankly, it was getting sort of boring. And so I said to David, why don't you go look for a gene that's important for development, that's only found in the eye, and that is conserved in biology, and that nobody else is working on. So he did a computer analysis of such genes. There were about 13,000 of them in the gene database at the time. And he found one out of the 13,000 that fit the criteria he and I had set out. And so the database indicated that this was an eye-specific gene, but as soon as he started to study it, it was clear it was actually more expressed in the rest of the brain than in the eye. But by that point, it was clear it was so interesting, I wasn't going to let it go. Yeah, you know, it was related to other genes that were important for development and that were located in synapses, and it was novel and nobody was studying it. So David worked on this project, in fact, for about 7 years. PhDs take a long time. And he actually stayed. He met his wife, who was another graduate student in my lab. And so he stayed for an extra 2 years doing a postdoctoral fellowship. And so this gene we discovered, NETO1, is a protein found in synapses, and it helps one of the major proteins that's involved in sending signals between nerves. It facilitates the function of that protein. And
It's involved in learning
and memory. Yeah.
Yes.
And when we got rid of this protein called NETO1, the learning and memory functions were impaired in a very subtle way.
Okay.
So that was nice. But the cool thing was we found we could compensate with a drug for that learning defect. So it was one of the first examples of showing you could correct an inherited learning defect with a drug. Okay, so it's a nice principle to establish. And of course that leads all kinds of people with damaged children and learning defects, you know, it lends them a kind of unrealistic hope that we can do something for such children in the short term. I think in the long term it shows the principle that you can do this. And that principle has now been established for about 15 in animals, okay, yes. in animal models for 15 inherited kinds of learning defects. So I think it's kind of a wonderful achievement that this has been discovered, because I think we all thought that if you have an inherited learning defect, you're set in stone and you don't go home.
Yes, exactly.
And this shows that isn't necessarily the case.
Yes.
And so I think the marriage of genetics and neurobiology is going to bring this to medical reality and hope, you know, gradually, and it won't be able to help everybody. It'll be able to help some individuals for sure in the future.
This is very exciting.
But there's another part of the story I want to finish.
Yes,
so I mentioned that David Ng met his wife in my
Yes.
lab. So then he's now at Columbia University training with one of the world's leading neuroscientists named Tom Jessel. But anyway, they got married, and they came to my door. They were leaving for New York in January of some years, about 3 or 4 years ago. And in their hands, they had a baby and 2 bound PhD theses. So that's the best output I've ever had from my lab.
That's wonderful.
What is the direction of your research at the moment?
I've got a good problem that's tractable, that's soluble, and tackling a problem that may be insoluble. And so the insoluble one may be insoluble, to be realistic, at least this point in time, unless the technology advances, is the following. So one of the areas I study are inherited retinal degenerations. Okay, so these are a group of diseases that are very much like having Alzheimer's of the retina. So in other words, what I mean by that is the retinal cells, the photoreceptors, work well for years or decades,
And then give out.
and then all of a sudden they don't work. And these are single gene diseases like cystic fibrosis and muscular dystrophy, where if you get the bad gene, you're going to get the disease. Okay.
Right.
So, but the mystery in these diseases, and in all inherited neurodegenerations like Huntington's disease, you would have heard of, and amyotrophic lateral sclerosis, and terrible diseases like this. There are inherited forms of Alzheimer's and of Parkinson's, you may know. So the mystery in these diseases is the following. So neurons are referred to as being born,
Yes.
okay, and then they normally live forever, you hope, if you don't drink too much, and then they die with you. But in these diseases, the peculiar thing is the mutation is present from birth in the neuron. So the neuron is born with the mutation, this mutant neuron. But oddly enough, it functions well for years or decades, and then it dies prematurely. So it's a great biological mystery, how it can be born and function normally for years or decades and then die. So there's a big series of question marks about what happens between the birth of the neuron and its death years or decades later.
Would this relate to macular degeneration? Is that something else?
There are some, well, I'll get to that, but there are some single gene inherited forms of macular degeneration. You're referring to more garden variety of macular degeneration, which is an epidemic in Western countries. Let's come back to that. Okay. But these single gene diseases, the question mark is between the birth of the abnormal neuron, the mutant neuron, and what's going on inside the cell when it's seeing normally or thinking normally or whatever it's doing for those decades. What leads it suddenly to die? So that's the question we're trying to address using mouse models of these genetic diseases of the eye, which are actually fantastic, perfect models of the human diseases, because eyes are pretty simple. The genes do the same thing in humans and mice. And when they're messed up with, you get the same kind of abnormalities in mice and humans. So the mice are wonderful models to study. But trying to figure out we're using sophisticated current biochemical and genetic methods to try to look at how what's abnormal in that cell that predisposes it to its premature death. The one thing we found that is counterintuitive is it's not advanced, accelerated wear and tear, which is what you think it might be.
Right.
The cell has the mutation, and the neuron therefore ages faster. In these eye diseases, that is absolutely not the case. That's the one thing we've discovered, but we haven't discovered the biochemical basis of what's going on. So, and it may not be possible to do so. So to play it safe, we have a more predictable problem, which is we found, I won't go into the details, but we found a gene that's important for eye and brain and lung development. It's perfectly uncharacterized. Virtually there's one paper on it in the literature. Right. And so what we're doing is, this is obviously an important gene. If you get bad lungs and bad brains and bad eyes, you want to know what that gene normally does. So, and this is the kind of thing I've done many times before, and so it's good for graduate students and postdoctoral fellows, because they know it's something where they can get results, and so that's pretty exciting. And
are you doing that now?
Yeah, we're doing that now.
At Lady
yes.
Davis.
Yeah, but we're starting from scratch, so it takes a long time to get all the reagents and get rid of the gene and get mice that don't have that gene and see what happens, and so it's a slow project initially. So that's what we're doing. So going back to macular degeneration. Yes. So that's a Western. It's an epidemic in Western countries. About 30% of people over the age of 75 have macular degeneration, and a good fraction of them become blind.
Yes, of course.
And it's probably occurring in Western countries, not because of anything Western, but because people in Western countries, developed countries, live longer. So they live long enough to get it. Okay. The only known contributor before genetic studies began on it is smoking. So another reason not to smoke.
to
Okay.
smoke.
And we, you know, I'm on the scientific advisory board of an American organization started by a wonderful person named Herb Lotman,